You know what? I like your curly hair. You know, Hrithik Roshan's eyes are blue, but mine are black. How? Eye color, hair texture, skin color, from where does we all get these traits? These traits are inherited to us from our parents. Now you can see that traits are of two types, dominant and recessive. Dominant are easily visible, they are always shown. And recessive, you can say they are hidden or suppressed by the dominant traits. Let's read their properties. Dominance and recessive. A dominant factor or allele expresses itself in the presence or absence of a recessive trait. Dominance alleles are always expressed, but a recessive trait is able to express only if the dominant trait is not present or in the absence of a dominant trait. For example, we are going to learn about all this in this video. Class 10, Term 2, Heredity and Evolution, Genetics Part 2, learned by Chetna Sharma, and our channel's name is KiddyCam. Please like, share and subscribe. The examples for dominance are, for example, tall plant, round seed, violet flowers, etc. These are the characters of a pea plant. Recessive traits, dwarf plant, wrinkled seed, white flower are the recessive pea plant traits. The major difference is dominant alleles is denoted by the capital letter when we are going to do any type of experiment over dominant and recessive. And recessive alleles are denoted by the small letter. You can see that capital B is a dominant trait, small b is recessive, and capital B and small b will be the heterozygous. So in my previous video, I have explained about homozygous, heterozygous and hemizygous. Important terms to remember: backcross is a cross in which the first generation individuals are crossed with any of their parents. Test cross is first generation offspring is crossed with the recessive parent, in which the total generations obtained are 50% dominant and 50% recessive. Reciprocal cross when two parents in the experiment in such a way that A is female and B is male. In another experiment, what will we do? A will be male and B will be female. Punnett square checkerboard method. It was firstly used by Reginald C. Punnett, 1927. This method is the representation of generations to analyze in the form of symbols of squares. Male gametes lie horizontally and female gametes lie vertically. It depicts both genotypes and phenotypes of the progeny. Dominant have been shown with the capital B red color and recessive is small b blue color. Now this is the diagram of a chromosomes in which the red chromosome, you can see the capital P, small a and capital B gene loci. Capital B is dominant character and on blue small b is a recessive character. So what will be the genotype? If there is capital P, the homozygous dominant allele will be represented. If there is small a, it will be a recessive allele homozygous. But capital B and small b will be heterozygous allele. Now we are going to learn about the very important topic, which is Mendelian inheritance. The inherited traits are determined by the genes that are passed from parents to children. So the inherited traits are nothing but what we are carrying from our parents. A child inherits two sets of genes, one from each parent. As I have showed you in the sex determination cross, XY is for the male gamete and X is for the female gamete. So whatever we carry, it may be like if the female child have XX, then one X may be from the father and another X will be from the mother. A trait may not be observable, but its gene can be passed to the next generation. Each person has two copies of gene, one copy from mom and second copy from dad. These copies may come in different variations known as alleles that express different traits. For example, two alleles in the genes for freckles are inherited from mom and dad. Allele from mom has a freckle capital F and allele from dad has no freckle small f. Child has the inherited gene pair of alleles, which is capital F and small f, which is heterozygous. But we can see that a dominant character is always expressed. That's why a child will be having a freckles. Now look at this man. Gregor John Mendel. He is the father of genetics. Studied pea plants and performed experiments with the pea plants, and he used his mathematical approach to explain this. Gregor John Mendel is responsible for the governing inheritance of traits. Gregor John Mendel, between 1856 and 1863, Mendel cultivated and tested some 28,000 pea plants. He found that the plants of offspring retained traits of the parents. Site of Gregor John Mendel's experiment garden in Czech Republic. Types of genetic crosses. Monohybrid cross, cross involving a single character or a trait. It may be anything. It may be a flower color or a shape. Dihybrid cross, in which a cross is involved with the two characters. It may be any combination. Here is the example of the flower color and the plant height. Now let's study about Mendel's pea plant experiments. Why? But why only pea plant? These peas are called Pisum sativum. The reason behind choosing a pea plant is it can be grown in a small area. It produces lots of offsprings. Produce pure plants when allowed to self-pollinate several generations. Garden pea is known as a Pisum sativum. Number of the chromosomes it have 14. Phenotype is affected by the environmental factors. The steps involved is Mendel's experiment involved 4 steps as selection, hybridization, selfing, and calculations. His results led to the formation of the law of genetics later. Now we are going to select a pure plants, hybridization of the pure plants for the first generation, and self-pollination of hybrid plants for second generation and third generation. Mendel's experiment methods. Mendel hand-pollinated the flowers using a paintbrush. He could snip the stamens to prevent the self-pollination. Covered each flower with a cloth bag. He traced traits through the several generations. What is pure plant? A pure line is a plant or animal that is genetically pure for particular character and will give rise to small characters after self-fertilization or interbreeding. But how Mendel began? Mendel produced pure strains by allowing the plants to self-pollinate for the several generations. Now these are some dominant and recessive characters of the pea plant traits. Seed shape, round and wrinkled. Round is dominant and wrinkled is recessive. Seed color, yellow and green. Pod shape, inflated and constricted. Pod color, green and yellow. Flower color, purple and white. Axial. Flower location, axial and terminal. Plant size, tall and dwarf. Now let's see dominant and recessive. Seed shape, smooth is dominant and wrinkled is recessive. Seed color, yellow is dominant and green is recessive. Pod shape, inflated is dominant and wrinkled is recessive. Pod color, green is dominant and yellow is recessive. Flower color, purple is dominant and white is recessive. Flower position on stem is dominant, at tip is recessive. Stem length, tall is dominant and dwarf is recessive. Generation or gap. Parental P1 generation. The parental generation is a breeding experiment. F1 generation is the first offspring in a breeding experiment, or it will be called as a first filial generation. F2, the second generation offspring in a breeding experiment, or will call be the second filial generation. Following the generation, cross 2 pure plants between capital T and small t. The results in hybrid is capital T and small t. The cross 2 hybrids will get capital T, 3 tall and 1 short. So you can see the 3 talls will be, it may be homozygous or heterozygous, but short will be homozygous, small t and small t. Let's study about the monohybrid cross. The characters which we are taking for monohybrid cross is seed shape. We know that seed shapes are round and wrinkled. Round is dominant, so we are going to take capital R for round, and wrinkled is small, so we are going to take small r for wrinkled. We are going to do the cross between the round seeds and the wrinkled seeds. So the gametes for the round seeds is capital R, and wrinkled is small r. After doing the cross, what we are going to get is genotype capital R small r, phenotype is round, genotypic ratio all alike, and phenotypic ratios are all alike. P1 monohybrid cross review is homozygous dominant and homozygous recessive. Offsprings all were heterozygous. That is called first generation, first allele generation, and genotype and phenotype are all alike. So you can see this is being showed the ratios also. The genotypic are capital R, capital R, capital R, small r, and small r. Phenotype is round and wrinkled. Genotypic ratio will be 1:2:1, and phenotypic ratio will be 3:1. What is genotypic ratio? We can separate them on the basis of their genes. But phenotype, physically we can see that they were all round. So the round were 3 and wrinkled were only 1. That's why phenotypic ratio is 3:1. You can see it is 25% homozygous, capital R, 50% heterozygous, and 25% homozygous recessive. You can see in this figure how does a P look like. You can see that few are rounds and few are wrinkled over here. Thank you for watching this video. Like, share, subscribe, and hit the bell icon to get notifications.